A-Level Biology 9700, Paper 4, Variant 2, October, November 2024, question number 3 and 4. And first of all, we will discuss the question number 3, which is very simple question. The treatment of many diseases has been improved by the use of genetic technology in medicine. So explain what is meant by genetic engineering. So genetic engineering, also known as genetic modification. Modification means the modification of DNA, the modification in the DNA. So is the process of altering. How can we modify the organism, or how can we modify the DNA of an organism? By the process of altering the DNA makeup of an organism, this can improve changing a single base pair. Single base pair means we can change adenine. There are 4 nitrogen bases: uh, adenine, thymine, guanine, and cytosine. These 4 nitrogen bases are involved in the formation of DNA molecule. So base pair means when any pair, that is adenine always combined with thymine and guanine always joined with cytosine. So removal of any base pair, we can change it. We can delete a region of DNA. We can delete a region of DNA. A small segment of DNA can be removed. A new segment of DNA can be inserted. Why do we need to remove that DNA, which is faulty, which is unhealthy? So we can remove that faulty DNA segment we can replace it with the healthy segment of DNA. The goal is to modify the organism's characteristics in a specific way. So our goal is in the modification of the organism. So in this regards, we can say that the genetic engineering is the process of directly modifying the DNA of an organism to change its characteristics or traits. So the different techniques can be used for this purpose. How do we change the DNA? Now, first of all, the first step which can be involved in the change to make change. First of all, we can edit the genes, gene editing. Gene, the first technique is gene editing. By gene editing, we can make precise or changes to an organism's genome. And the second technique can be used, gene cloning. And gene cloning, copying. Cloning means copying. So the copying genes to the inserted into the other organism. And number 3, recombinant DNA technology. Recombinant DNA technology. And what is recombinant DNA? Recombinant DNA means combining DNA from different sources into one molecule. So that will be the DNA which will have the characteristics of different molecules. So it will be a recombinant DNA. So these are the different techniques with the help of which we can replace, we can insert, we can change the DNA of an organism. And this genetic engineering is involved in different fields. As for example, the number 1 fields in which medicine. For medicine, we can make insulin by this method, gene therapy. So the second field is agriculture. Agriculture, we are creating pest resistant, insecticide resistant, or drought tolerant crops. Number 3 is we make use of this to study the gene functions. We are making use of research function to study gene functions. So in this way, genetic engineering is involved in many fields of our life. Now, the B part of this question is Leber congenital amaurosis is an inherited eye disease in LCA. The photoreceptor cells in the retina die at an early stage. This causes impaired vision in children, which can progress to blindness. Mutations in different genes cause different forms of LCA. One form of this disease, LCA2, it means Leber congenital amaurosis 2, is caused by a mutation in the RPE65 genes. Gene therapy has been used to treat LCA2. So LCA2, so outline how an inherited eye disease such as LCA2 is treated with gene therapy. So here, first of all, the question is, what is gene therapy? In the previous part of this question, I had discussed that is the genetic engineering can be done by using different techniques, and one of them was genetic therapy. So the gene therapy is medical approach that aims to treat or prevent disease by correcting underlying genetic problems, often by replacing a faulty gene with a healthy one or introducing a new gene into the body. So gene therapy, how do this done is can be done? That is by uh, replacing of a faulty gene, or introducing a new gene into the body. Replacing a faulty gene or introducing the new gene. So gene therapy can be done in this way. So how an inherited eye disease is treated with gene therapy? Now we have to discuss what will be done in this regards, how this gene therapy can be done. So here I have already written the answer for you, because sometimes you feel most of the students complain me that your handwriting is not clear. So that is the reason I already typed and then I will explain the question. So answer the question, the gene therapy of for LCA2 is called Luxturna and Luxturna. Uh, First of all, and Luxturna uses a harmless adeno-associated virus as a vector to deliver the normal human gene RPE65 directly to the retinal pigment epithelium cells. Luxturna is administered through a one-time surgical injection under the retina in each eye, and this procedure ensures that the gene reaches the target RPE cells. The delivered normal RPE65 gene begins to produce functional RPE65 protein. So I would like to explain all this. What is Leber congenital amaurosis? Is an inherited eye disease, which we have read. And this is caused by mutations in the RPE65 gene. Mutation in RPE65 gene. So this gene provides instructions. Actually, what is the function of this gene is to provide instructions um, for making a protein essential for normal vision. So specifically within the retinal pigment epithelium, RPE. RPE is actually the retinal pigment epithelium. So a retinal pigment epithelium 65 gene. So the cells at the back of the eye. So this retinal pigment epithelium is at the back of the eye. this RPE65 protein. So the responsible protein is 65, which plays a crucial role in the vision cycle. What is vision cycle? A process that converts light. So vision cycle. What is happening? Vision cycle. The protein responsible for this is RPE65. So what is happening? What happens actually? The RP proteins, vision cycle, light energy is converted, light is converted so light energy converts light entering the eye into electrical. So light energy entering the light into the into electrical signals. And then these electrical signals are sent to the brain. So when the RPE65 gene is muted, the RPE65 protein either does not function correctly or is not produced at all, disrupting the visual cycle and leading to severe vision impairment from early childhood. So what is happening? What is this cycle? That is uh, the normal functional vision takes place by the retinal pigment epithelium, which is present at the back of the eye, and the RPE65 protein plays a crucial role in the visual cycle. So what is visual cycle? This is a process in which that converts light, the light which is entering into our eyes, into electrical signals. So light entering, I think I must be very clear over here. The light entering, the light and which is entering into our eye is converted into electrical signals, and these electrical signals are sent to the brain. So this is the actually the visual cycle. So due to mutation, this visual cycle is not properly working. So it will be disrupting, and it will lead to a severe vision impairment from early childhood. So gene therapy for LCA2 aims to replace the muted RPE65 gene with a normal functional copy. So the currently approved gene therapy for LCA2 is called Luxturna. Luxturna, which I have written over here. So this Luxturna, in which a harmless adeno-associated the virus, which is very harmless. So this virus is used as a vector to deliver the normal human gene. We have to replace the RPE65 to the retinal pigment epithelium cells, RPE. So here RPE epithelial cells to Luxturna is administered. So this Luxturna is administered through a one-time surgical injection under the retina in each eye. Under the retina in each eye at a time. So one time surgical injection is under the retina in each eye. The two eyes are treated separately. Two eyes are treated separately with at least 6 days between procedures. Both eyes cannot be treated at once. So the both two eyes are treated separately with at least 6 days between procedure. So the gap should be of 6 days. So minimum should be 6 days. So the precise delivery method ensures that the curative gene reaches the target RPE cell. So with the help of their injection, surgical injection, the normal human gene RPE65 is injected So Luxturna is administered through a one-time surgical injection each eye, and this procedure ensures that the gene is reaching the target RPE cells because protein is needed over there. The delivered normal RPE65 gene begins to produce functional RPE65 protein. So the we can say the newly produced functional RPE65 protein can then participate in the visual cycle. No, it will, this newly formed will participate in visual cycle. So visual cycle already too, when light enters into our eye, that light is converting into electrical signals, and those electrical signals are sent to the brain. So this is the visual cycle I have told you again and again. So this cycle is disrupted when there is mutation in the protein RPE65. So due to which the blindness occur in the at very early age. So after this restoration, you can say by restoring a key step in the visual cycle, Luxturna can improve the vision of individuals with LCA2, particularly their ability to see the low light conditions. So clinical traits have shown improvements in functional vision and light sensitivity in parents treated with Luxturna. One more thing, students, it is important to note that Luxturna is indicated for patients with confirmed biallelic, both copies of the genes are muted. In case Luxturna is applied when both copies of genes are muted. So RPE65 mutation associated retinal dystrophy who have visible retinal cell. So the therapy aims to halt the progressive retinal degeneration associated with LCA2 and stabilize the remaining vision. So this is what, that is, the cycle is restored by replacing the RPE65 gene, muted RPE65 gene with the normal RPE65 gene by the help of gene therapy. Next question is, suggest why the eye is a suitable organ for gene therapy. Well, students, this is the answer of one mark. The you know that eye is a relatively self-contained and accessible organ. So it can be. Let me write. Eye is first of all self-contained and accessible organ. So it can be easily reached for direct injection of gene therapy vectors such as subretinal injections for retinal diseases. This localized delivery minimizes the potential for the vector to other spread to other tissues. So so in this way, and second is immune privilege. Immune privilege. So the eye has a degree of immune privilege, meaning that the immune system is suppressed within the eye compared to other tissues. So this reduced immune response is beneficial for gene therapy because it lowers the likelihood of body rejecting the introduced genetic material or the cells that has been taken it up. And the third reason for this suggesting why the eye is suitable organ for gene, that is compartmentalization. So what does it mean? The eye small size and compartmentalized means structure allow for precise delivery and containment of the gene therapy vector very easily. So, and then you can see I have written the changes in vision, and the number 4 option of the answer, changes in vision and the health of retinal cells can be directly and non-invasively monitored using various ophthalmological techniques. So what are these these are the actually uh, electro retinography, visual acuity tests, or optical coherence tomography. So these are the different techniques which can be used for this, easily apply on this. They allow clinicians to easily assess the effectiveness of the gene therapy. They can easily assess how do this gene therapy is successful, and you can see, or it is not successful or successful. So other you can say uh, other one more option can be written, but you can choose any one of them. So post mitotic cells. Many of the target cells for gene therapy in the eye, such as retinal pigment epithelium and photoreceptor cells, are post-mitotic, meaning they do not actively divide. This is advantageous because the introduced therapeutic genes are more likely to persist in these stable cells over the long term, so potentially leading to a lasting therapeutic effect. So these are the very different, you can say, eye self-contained and easily accessible organ. Secondly, immune system, privilege, compartmentalization, and the fourth one I have written over here, and 5, and this is the sixth one. So I these are the 6 options. You can write any one of them. So the answer is of 1 mark. Now next is LCA 10. In the previous it was LCA 2, now it is LCA 10. Is a different form of LCA caused by a recessive mutation in the CEP290 gene. So this is the CEP290 gene. This gene codes for the protein CEP290, which is involved in the correct functioning of the photoreceptor cells in the retina. So these are the different genes responsible for the mutation is a CPE22 causes an error to be made with the primary transcript in place to form messenger RNA. The abnormal messenger RNA that is formed has an extra sequence of RNA nucleotides known as exon X. So between exon 26 and 27, exon X contains a stop codon. Compares the effect of the mutant. You can see over here normal and LCA10 section of DNA of CE290. And what is the primary transcript? Intron, and you can see LCA10 there is mutation. Due to mutation there is an extra. EXONX is an extra. You can see this one. So due to which CEP, this is the normal polypeptide and it is shortened CEP290 polypeptide. So in 2022, research was carried out into possible treatment of LCA10 using genetic technology. A human clinical trial investigated a treatment of LCA10 using a short RNA nucleotide sequence known as uh, sepofarsen. Sepofarsen binds to the section of the primary transcript containing the CEP290 mutation so that normal splicing occurs and functional CEP290 protein is synthesized. People in the clinical trial received regular treatment with cepofarsen to the eye with the greatest loss of vision treated eyes for a period of 12 months. Changes in the light perception, visual acuity, of both eyes were measured using a vision chart. A negative change in the visual acuity scores how shows an improvement in visual acuity. So here you can see the graph. Impaired acuity, it is increasing in the downward, that is improved acuity. So the time is of 12 months, and both of them, I not treated and I treated. In not treated, you can say the upper one is not treated and the lower one is when treated. So lower one shows the improved equity, visual equity, whereas the which was not treated, it shows the less improvement. Now describe the results of the clinical trial data shown in 3.2. I have told you that the results of the clinical trial data shows. So here you can see the first of all, we are moving towards the image one. Image one. So uh, you can see over here LCA 10 and there is normal. And then we are moving to, on graph. We will discuss figure 3.2. So we have to discuss the second one. Error. In here you can see in the graph. First of all, figure this image line graph showing the results of the clinical trial data monitored in the first image. So here you can see over here, that is initial increase in impaired equity, a dip in the graph shot after the start. So SEPA formation sustained improvement in the when there is treatment, whereas there is an initial increase in impaired equity. Let me move towards the graph, then you will understand it. First of all, there is this is the dip which is not treated with, and then visual acuity improves considerably, reaching a peak around 3 and 4. Here you can see little bit improvement is there. You can see this the fluctuations. You can see the fluctuations over here in the down, little bit improvement, then it is going in the upper direction improvement. But here you can see a consistent improvement when it is treated. You can say the visual acuity improvement in this way, but here in 8th and 9th month, little bit this improvement has been shown in this graph. So figure 3.2 provides visual evidence uh, evidence suggesting that the ciprofloxacin treatment resulted in a noticeable and relatively sustained improvement in visual acuity in the treated eye compared to the untreated eye control in patient with LCA10 due to the specific splicing mutations in CEP290 gene. This supports the potential of the gene therapy approach for treating this form of LCA10. So in this way, the appear slight, you can say, changes. And due to these changes, we can say that the there is a support, the potential of the gene therapy approach for treating this form of LCA10. So here in this graph, you can easily compare these two lines. And untreated eye and treated eye. Untreated may you can see the visual acuity in the untreated eye generally shows a slight initial improvement as I have shown to, and This is the little improvement, and here shown improvement. And then it is again, you can see the fluctuations. You can see the fluctuations. And on the other hand, the treated eye with cepaforsen, the usual is the treated eye shows a more significant and sustained improvement. This is the this is the sustained improvement. You can see, and as comparison, you can easily compare both of these graphs. So little bit, you can say, less improvement has been shown over these months. So in this way, you can see these two graphs are helpful to compare both type of curves. Now the next question. Another method being investigated to treat LCA10 is to use a gene editing tool known as the CRISPR CRISPR Cas9 system has uses a short length of RNA called guide RNA. Guide RNA is complementary to the target RNA and is linked to a nuclease enzyme called Cas9. Cas9 breaks phosphodiester bonds in DNA. You know, the bonds between DNA molecules are phosphodiester bonds. So the cell repair mechanism repairs the cut in DNA after the modification has taken place. A vector delivers Cas9 and two specific guide RNAs to the phosphoreceptor cells. They act on the section of DNA which contains the mutation. Exon X is no longer added to the messenger RNA. Explain how this method used to treat LCA10 is an example of gene editing. The CRISPR-Cas9 system is used. So explain how this method used to treat LCA is an example of gene editing. Actually, we have to discuss about gene editing over here. So editing means there will be addition into and CRISPR Cas9 system is a powerful and precise tool for making changes to specific DNA sequence within a cell. So it essentially acts like a molecular cut and paste system. So molecular cut and paste. Molecular cut and paste system. So the key components for this are gRNA, guide RNA. So this is the RNA that is designed to be complementary to a specific target DNA sequence in the genome. In the context of LCA10, the gRNA would be designed to match the RNA region containing the mutation in the CEP290 gene. So Cas9 enzyme. Second is Cas9 enzyme. This is nuclease, an enzyme that can cut DNA. So this is associated with the gRNA. The gRNA guides the Cas9 enzyme to the specific target DNA sequence. So they will target the DNA sequence, and then there will be the once the gRNA guide Cas9 to the correct location on the DNA, and then the Cas9 enzyme creates a double strand break in the DNA at that precise point. So CRISPR is used to target specific section of DNA. So specific section of DNA. that contains the mutation responsible for LCA10. So the point, so this is the specific section of the DNA or the point where there is mutation for LCA10. So a guide RNA directs the Cas9 enzyme. RNA will direct, it will give instructions to enzyme to location of the mutation. So Cas9 then makes a cut in the DNA at the location. So Cas9 will act as a scissors. So it will cut in the DNA at that location and the cell's natural repair mechanism will fix the cut. So that cut, at that cut, there will be the natural reparation during which the mutation can be removed or corrected. Muted part can be corrected or muted part can be removed. As a result, exon X is no longer added to the messenger RNA, so which prevents the production of faulty protein. So this exon X is responsible to produce the faulty protein, so it can be reduced, so it can be prevented in this way. So these you can say in this way you can see uh, exon X is no longer added to the messenger RNA, so due to which, while it does not explicitly mention the intron based on the previous image. So in this way, we can say all this function would allow for the production of a functional CEP290 protein. So this is an example of gene editing. So this method is a clear example of this gene editing because it involves making precise alterations to the genetic material or DNA, or specifically target modification is over there. You can say the CRISPR Cas9 allow for highly specific targeting of a particular DNA sequence uh, at the site where the mutation is there. Then direct manipulation of DNA. The Cas9 enzyme directly cuts the DNA at the targeted location and then altering gene expression by modifying the DNA sequence through small insertions, deletions, or a precise, you can say we can insert, we can delete. So the CRISPR-Cas9 system aims to correct the underlying genetic defect that is causing the disease. So in this case, it aims to restore the correct processing of the CEP290 genes RNA leading to the production of functional protein, faulty protein to functional protein. So we can say that the CRISPR Cas9 provides a way to directly edit the faulty gene in the patient cells, offering the potential for a more permanent therapeutic effect compared to other approaches like RNA-based therapies that might require repeated administration. So it was all about this. You can follow the answer. Now, question number 4. Genetic crosses can be used to investigate patterns of inheritance. So, a mutation is a gene involved in fruit color in tomato plant. Solanum lycopersicum results in the production of yellow fruits instead of red. Red fruit and yellow fruit. So due to mutation, yellow fruits will be produced. A genetic cross was carried out between a pure breeding plant with a red fruit and a pure breeding plant with a yellow fruit to produce the F1 generation. All offsprings plants produced red fruits. The F1 plants were then crossed with each other and the seeds produced were planted to obtain the F2 generation. Construct a genetic diagram to show the cross of F1 generation that produced this F2 generation. So first of all, I will show you how there is production of this F1 generation. So for this purpose, we pure purebred, purebred red, little bit move red fruits and yellow fruits, muted yellow fruits. So I am moving little behind. Capital R will be used for this purpose. So capital R will show the red fruits, and small or small will show the yellow fruits. No, all the offsprings of this cross have one produced red flowers. So how? A pure breeding red fruit was crossed. Pure breeding which always give the red color. So red fruits, red into yellow. So parents, these are the P1 phenotype. And genotype of the parents is capital R, and small r. And gametes, and F1 is capital R, small r. So now we have to get the F2 by F1 into F1. So F2 can be, first of all, in F2, the phenotype of parents is red into red from F1, and genotype is capital R, and small r. So by making by making by making use of Puneet's scale, we can have capital R, capital R, capital R, small r. So in this way, we can see over here gametes. Now we will make the gametes capital R, capital R, small r. So after Puneet's scale, you can see the phenotype. They will be 3 red, ratio 1 yellow. And genotype, one is to 2:1 ratio. So we can say the genetic diagram above shows the cross of the F1 generation illustrate how the F2 generation with a 3:1 phenotype ratio of the red fruit is produced. So it was all about this question. Moving towards it was monohybrid cross. Now the next is a theoretical dihybrid process involve two genes located on different autosomes. Each gene has two alleles, one dominant and one recessive. A parent homozygous dominant for both genes is crossed with a parent that is homozygous recessive for both genes. This produces F1 individuals that are then crossed to produce the F2 generation. So state the phenotypic ratio of the dihybrid F2 generation and explain why some of these offspring's phenotypes are different from the original parent phenotypes. So here you can see the typical phenotypic ratio of the F2 generation in a high dihybrid cross involving two independent assorting genes with complete dominance is 9 is to 3 is to 3 is to 1. As you know, in monohybrid cross, there is only one characteristic which has to be observed, and they produce the four uh, genes. But in this case, you can see uh, there are two dihybrid cross is there. So two genes are involved. So due to two genes, the ratio becomes, instead of 4, it will be 16. And the ratio among them will be 9 is to 3 is to 3 is to 1. So for this, you can say the homozygous dominant parent is crossed with a homozygous recessive parent. For this purpose, to prove this 9 is to 3 is to 3 is to 1, you can see that we can capital A, capital A, capital B with, we can cross it with small a, small b. So in the all, you can say in the F1 will be A, A, B. So by making use of this as A genes, and then you can see um, then F2 generation, when the F1 individuals are crossed with each other, their gametes will carry all possible combinations of alleles. You can say F2 can be F1 into F1. So with this, you can see by making use of this A, you can see over here, the gametes formation will be in this way. So you will make use of Puneet's care in this way. A, A, B, A, B, A, capital B, A small. So in this way, you can have 16 combinations: 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16. And the ratio among their results will be 9 is to 3 is to 3. And 9 offsprings, they will show both dominant phenotypes, that is, which was of the parents phenotype. As for example, capital A, capital A, capital A, capital A, 9 will be, let me write over here, 9 will have this phenotypic ratio in this way. Or and 3 will have And then next three will ratio will be small a, capital B, capital A, small a, capital B, and small b. And one ratio will be small a, small b, which will be homozygous recessive parents. So this will be the ratio 9 is to 3 is to 3 is to 1. Now, explanation. I have explained this, how it will be produced. This ratio will be produced. So during meiosis in the F1 individuals, the homologous chromosomes carrying the different gene pairs separate independently. So this independent assortment leads to the formation of gametes with novel combinations. Novel means new combinations of alleles that were not present in the original parent gametes. When these novel gametes combine during fertilization, they produce offsprings with phenotypes that differ from either of the original parents. You can see the original parents. These were the original parents. As a result of meiosis, you can see now you can have these different types of combinations. You can have these different types of combinations which are not exactly the same with the parents. Many different combinations has been produced. As for example, only this combination and uh, only this combination and this combination resembles with the parents. All other combinations are different. And these combinations are produced as a result of meiosis. And you know what happens in meiosis? You know, in prophase 1 of meiosis, what happens? The crossing over takes place. Crossing over takes place in which the homologous chromosomes come close to homologous homologous chromosomes, they come close to each other. When they come close to each other, they exchange their parts. They exchange their parts, they are joined together. At metaphase, they are at the equator. In metaphase, they arrange themselves at the equator. In this way, they are joined together, but in anaphase, they get separate from each other. At anaphase, they separate from each other. And in this way, the each daughter cell will have the different combination of genes. So this is the reason that in F1 and F2, the different, uh, you can say, the offsprings phenotypes are different from the original parents' phenotypes. So it was all about today's lesson. So please like, share, and subscribe my channel. Thanks for watching. I always give my email ID in the description. You can send me a mail if you have some problems in the answers or in the question or in your understanding. Thanks for watching.